Assalamu alaikum. A warm welcome to all of you. We are glad to have you all here with us on International Brain and Biomedicines Conference IBBC 2023. I am Tahreem Iman, PhD scholar from Department of Physiology, Government College University, Faizabad. I am going to moderate this session. Today we have many informative talks on different fields of science such as molecular biology, bioinformatics, biotechnology, and genetic engineering. Let's make a start of today's session. I would like to invite Asia Siddiqa from Fuhad University of Science and Technology. She is going to present her research work with the title Genetic Characterization of a Family with Autosomal Recessive Bardet-Biedl Syndrome.
Welcome. I'm Asya Siddiqa, and today the topic of my presentation for IBBC 2023 is Genetic Characterization of a Family with Autosomal Recessive Bardet-Biedl Syndrome. So these are the outline of my presentation. First, I will discuss the introduction, then methodology, then results, conclusions, recommendations, and references. So first will be the introduction. It is an hereditably clinically heterogeneous, pleiotropic, and multisystem disorder. It has many phenotypes, showing many phenotypes characterized by the progressive retinal degeneration, polydactyly, obesity, renal irregularities to cognitive impairment. But it is also showing many other secondary medical conditions. It is a rare disease and estimated frequency is 0.7 in 100,000. And to date, 26 genes is reported to cause the BBS in diverse societies. And it exists in autosomal recessive pattern of inheritance, but also it is possibly inherited as an oligogenic trait. Second is methodology. First, what we do is that we take the informed consent form from the guardians of the patients. Then we do this blood sample collection. After that, what we do is the genomic DNA extraction, and it is using the same standard phenol-chloroform method. After that, the whole exome sequencing and data analysis. And after that, what we do is that we design the primers for the pathogenic variants, and we run the PCR for the amplification of the specific sites. And then we perform this we do the PCR amplification, and then we perform the segregation analysis through Sanger sequencing to check the variant is segregating in a family or not. Research objective first was to investigate the genetic cause of BBS in a family from district cohort, and then second was the important objective was to develop the genotype-phenotype correlations among the family members. So these are the results. First, I'll show you the family pedigree. In this, all the members, the affected, normal parents, all are present in this family pedigree. Whole exome sequence data analysis. How we conclude this data is that we use different filters, like we use the genotyping, genomate frequency, coding effect, variant type. Like all of these filters are used, and at last we come to the conclusion. And these are all used to filter out the variant, the genetic or the causative variant that is causing the disease. So here we have found, after the using different filters, we have found our variant, that is a BBS7, and which is causing a non-frameshift deletion. So the gene is the BPS7. Mutation is at there is a sequence, and this whole exome sequence analysis show that the BPS gene is showing a 3 base pair deletion at the nucleotide position 580 to 582, and 3 base pair deletion removed an evolutionary conserved alanine codon 194, which most likely altered the secondary structure of the BBS7 protein. The genomate frequency is 0. mutation tester is showing that is a disease causing. Franklin is showing it as a variant of uncertain significance. Versome is also showing it as an uncertain significance. This I have told you before that about the mutation tester. You can see here the results of the mutation tester. These are the results of the Franklin, which is showing it as a variant of uncertain significance. And this is the Versome, which is showing it as an uncertain significance. So now come towards the Sanger sequencing and segregation analysis. So as we have come to the conclusion that is a variant is B in BBS7, which is like causing the disease, which is responsible after we do the whole exome sequencing of a abnormal family member. So now we see that segregation in the families is happening, or in what kind of situation is that. So let's come toward that. So first, there is a family member which is individual 2-1. 2-1 sequencing result showed it as an heterozygous, because here you can see there are two peaks. So it is showing it as clearly showing it as an heterozygous, and we come towards the conclusion that it is a carrier for this genetic variant. And the individual 2 was also at the same position. It is genetic. It is also having two peaks, representing two peaks, and it is also heterozygous for the genetic variant. And then we'll come towards the individual 2-3. It is also showing the same situation is going on. It is heterozygous for the genetic variant. You can see clearly from the peaks that is showing double peaks. But the individual 2-4, you can see here the position. At that position, there is one peak, and there is no it is having the sequence. There's a GCT. You can see here, and it is having a single peak, and it is not neither it is heterozygous, neither is abnormal. It is a normal sequence. And the abnormal and sequence I'll show you on the next slide. The 3-4 individual is abnormal. 3-4 individual is also heterozygous at the same position. You can see here. And the individual 3-16 is also heterozygous for the same situation. And 3-3 is abnormal here. You can see here at the arrow that I'm showing you is that at this position there was a GCT that is deleted. This 3 codons letter, they are deleted. And also at 3-5, you can see here at this position there is a deletion occur. So now I'll come towards the conclusions and recommendations that the genotype-phenotype correlation established in the present study, it may improve the clinical diagnosis of affected individual for the better management and counseling, because it is very important to do it, because it is not only affecting one person or one individual, it is affecting the whole society. These are the differences. Thank you so much for your attention.
That was a really remarkable presentation. Our next presenter is Muhammad Fizan Ilahi Bhatti from University of Veterinary and Animal Sciences, Lahore. His topic of presentation is prevalence of hepatitis E virus infection among patients with clinical gastroenteritis in Lahore, a neglected public health issue.
Assalamu alaikum. My name is Muhammad Fizan Ilahi, and I am doing MPhil in One Health from the University of Veterinary and Animal Sciences, Lahore. My topic of presentation is prevalence of hepatitis C virus infection among patients with clinical gastroenteritis in Lahore, a neglected public health issue. Hepatitis C virus is a viral food and waterborne infection, primarily affects the liver and causes gastroenteritis. It is commonly found in developing countries. Its overall case fatality rate is low, but higher risk for pregnant women. Mortality rate for pregnant women in the third trimester is 10 to 30%. The objective of study are estimate the prevalence of hepatitis E among individuals with gastroenteritis-related symptoms in Lahore, Pakistan. Investigate the distribution of hepatitis E cases among different age groups and genders. Identify high-risk age groups for hepatitis E infection in the study population. Provide insights to inform future research and interventions for hepatitis E control in Pakistan. Study design. The total study period was 1 year, and it was a cross-section survey, convenience-based sampling, analysis of hepatitis E prevalence, age distribution, and gender distribution. For data collection, a questionnaire was utilized to gather information from patients regarding their name, address, age, gender, and disease status. Data was collected from 65 individuals, in which 32 were males and 33 were females with gastroenteritis-related symptoms. All statistical analysis were performed with R. For categorical variables, chi-square was used, and for numerical variables, t-test was used. Results are: total 18 cases were positive for hepatitis E, and overall prevalence was 27.7%, as 18 out of 55 cases were positive. 3 out of 18 cases were young, and 15 out of 18 cases were adults. So, prevalence in young was 16.67%, and in adults it was 83.33%. If we talk about gender distribution, there were 50% males, 9 out of 18, and 50% females, 9 out of 18. Findings: hepatitis E is actually present in the population studied. Adults are at a higher risk for hepatitis E infection, equal prevalence among males and females. Results highlight the need for targeted research and interventions to control hepatitis E in Pakistan. There is a need for targeted public health interventions to reduce hepatitis E transmission. Importance of focusing on high-risk groups such as adults and pregnant women. Encourage improvements in sanitation and access to clean water to prevent infection. Inform future research on hepatitis E epidemiology and risk factors in Pakistan and similar regions. Conclusions: hepatitis E is prevalent in the studied population in Lahore, Pakistan. Adults and pregnant women are particularly at high risk. Equal prevalence among males and females. Study findings can inform targeted interventions and prevention strategies. Effective public health measures can help control hepatitis E in developing countries like Pakistan. Thank you.
Thank you so much, Fizan Bhatti, for sharing your reflective knowledge with us. Our next presenter is Sadaf Masood from Kohat University of Science and Technology. Her title of presentation is Effect of Plant-Derived Smoke Synthesized Silver Nanoparticles on Maize Seedling.
Assalamu alaikum. I am Sadaf Masood, and the topic of my presentation is Effect of Plant-Derived Smoke Synthesized Silver Nanoparticles on Maize Seedlings. These are the contents of my presentation. First of all, I am talk about introduction of nanoparticles. As you know that nanoparticles are getting a lot of attention from researchers due to their wide application in biomedicines, drug delivery, and agriculture. There are two ways to synthesize nanoparticles, that is chemical synthesis and green synthesis. The nanoparticles which is synthesized from chemical synthesis, they produce toxic byproducts which is really harmful to human and plants, and also they require proper handling and caring. While the nanoparticles which are synthesized from green synthesis have a lot of benefits, that is, they are eco-friendly in nature, cost-effective, non-toxic, non-lethal, and no threat to life. There are many types of nanoparticles, but silver nanoparticles are one of the most vital and fascinating nanomaterials among several metallic nanoparticles that are involved in different applications, that is electrical, thermal, and biological properties. In our research, we use plant-derived smoke solution because plant-derived smoke solution have a lot of advantages. That is, they increase metabolic activity, increase plant height, chlorophyll content, and leaf surface area. Objectives of my research was to synthesize silver nanoparticles from concentrated aqueous smoke solution, and also to investigate the effect of silver nanoparticles on maize seedlings. The objective has been adopted by the following materials and method. In materials and method, first of all, we prepare smoke solution. After smoke solution, we add that smoke solution with 1 ml of silver nitrate solution, and we transfer that solution in orbital shaker for 24 hours. After 24 hours, we visualize that solution in spectrophotometry. After visualization, we perform centrifugation for phase separation. After phase separation, we move towards characterization to characterize silver nanoparticles through SAM, XRD, and FTIR. In experimental design, first of all, we sterilize the maize seeds. After sterilization, we prime these seeds with silver nanoparticles and smoke solution for 24 hours. After 24 hours, we transfer that seed in sand culture for 15 days. After 15 days, we harvest these seedlings for physiological parameters. Physiological parameters contain germination, root, shoot, length. Now we're moving towards results. Characterization of silver nanoparticles. In characterization of silver nanoparticles, the SAM image of silver nanoparticles indicates that silver nanoparticles are spherical in shape. Now we apply these nanoparticles on maize seedlings. The germination percentage indicates that the lower concentration, that is 5 ppm, increases the crop yield as compared to higher concentration, that is 10 and 15 ppm. The same result was obtained in shoot and root length, and also in dry weight of root and shoot. Now the conclusion of my presentation was silver nanoparticles successfully synthesized from concentrated smoke solution. Silver nanoparticles improved seed germination and seedling growth as compared to smoke. These are the references of my presentation. And then end of my presentation. Thank you very much.
Thank you, Sadaf Masood. That was really an amazing presentation. Our next presenter is Maha Rahman from Kohat University of Science and Technology. Her title of presentation is Identification, Growth, Kinetics, and Evolutionary Pattern of Spirulina Isolate from Freshwater Bodies of District Kohat, Khyber Pakhtunkhwa, Pakistan.
Assalamu alaikum. My name is Mahar Eman. I'm a PhD scholar in Guwahati University of Science and Technology. Today, I'm going to present one of the objective of my PhD research work, that is on identification, growth kinetics, and evolutionary pattern of spirulina species isolated from freshwater bodies of district Kohat, Khyber Pakhtunkhwa, Pakistan. These are the outlines of my presentation, which includes introduction, objectives, methodology, results, and conclusion, followed by references. Before getting in detail, I would like to discuss a little about spirulina species. So spirulina is unicellular, alkaliphilic, photoautotrophic, and filamentous cyanobacterium, and generally characterized by regularly coiled trichomes. Spirulina have a quite valuable species, and it is known for its high proliferation rate, carbon dioxide sequestration, and high biomass yield. Here are the importance of some of the importance of spirulina species. So spirulina is popular for its antimicrobial activities, including antibacterial and antiviral activities. It is metalloprotective, immune stimulant, have antioxidant properties. It is anti-carcinogenic and boosts immune system. Nowadays, spirulina is getting worldwide attention as a dietary supplement for humans as well as for animals. It is also documented as generally recognized safe food by the United States of Food and Drug Administration. The World Health Organization considers spirulina as one of the greatest superfoods on earth. Also, NASA described spirulina as one of the greatest superfood, compact food to travel in space because a small amount contains a wide range of nutrients. So if we talk about the economic importance of spirulina species, so spirulina is the prominent species for dietary supplement worldwide, with worldwide production of 40 million USD annually, and it is estimated that by 2025, the global spirulina market is expected toward 629.6 USD million with a compound annual growth rate of 9.4%. To sort out the taxonomic issues, we find that 16S ribosomal RNA is the best marker because it has a central role in inferring phylogenetic relationships and identification of prokaryotes. This gene possesses most of the characteristics needed for a good phylogenetic marker gene, such as a universal distribution in prokaryotes, functional consistency, relatively large size. Here are the objectives of my presentation. So first one is identification of spirulina strain in Kohat freshwater reservoirs. Number 2, evolutionary divergence analysis. Number 3, optimal growth condition and growth kinetics. To fulfill the objective, we followed this methodology. So first of all, we collect the sample from the top and bottom surface of the water reservoirs of Kohat. So for this, we perform microscopy and then enrich the sample. After that, we transfer that sample to the lab aseptically. For isolation, we followed through two different methods. One is the pour plate method containing agar media with BG11, and the other one is that we only use BG11 media without agar. Then after each 24 hours, microscopy were performed to check the culture. For further confirmation of our species, We perform molecular identification using 16S ribosomal RNA marker and then move towards the growth kinetic experiment. So for this, we optimize the condition for comparative analysis at different pH, temperature, and light intensity. Here are the results. You can see in the map that we select the city of Kohad because Kohad is the main district of KPK, Pakistan, and the presence of limestone and heavy metal in the mountains of Kohad make the water more alkaline in nature, which is suitable for the growth of spirulina species. So we chalk out some of the water reservoirs of Kohad. Some of the names are mentioned here. That is Tana Dam, Bonachari, Jungle Hill Chashma, Thunder Dam, Jarma, Bhandiali Dam, Dhoda Sharif, and Chili Bagh. So here we mark the area of Chili Bagh, Kohad, red in color because the first report we get were from Chili Bagh, Kohad. This is the area in Ustarzai. Here is the microscopic picture of spirulina species. Here are the results of DNA isolation and PCR product amplification. This is the phylogenetic tree for spirulina species. Here are the growth optimization results. So growth of spirulina in response to varying temperature condition. So the best results shown were at 20 degrees centigrade. Here is the result of light intensity, growth of spirulina in response to varying light conditions. So the best results were shown at 2400 lux. And growth of spirulina in response to varying pH conditions. So the best results shown were at pH 10. Here is the result of growth rate calculated from measuring optical density and weight of spirulina species per ml for DG11 media for a period of 12 days. So the maximum growth we get were at day 10. Here are the conclusion of my presentation. So first one is that spirulina is a non-toxic balanced diet with high concentration of protein, vitamins, and essential fatty acids. Number two, the development of spirulina in the lab has been investigated under various settings. Number three, the best spirulina growth yield was achieved at pH 10, temperature regimes of 20 degrees centigrade, and light intensities of 2400 lux. The experimental results allow for accurate anticipation of biomass production for large-scale spirulina cultivation with higher photosynthetic content. Here are the references of my presentation. I cordially acknowledge and gratitude to Professor Dr. Rehan Naeem, my supervisor, Professor Dr. Noor Muhammad, Chairman of Department of Biotechnology and Genetic Engineering of Kohad University of Science and Technology, and special thanks to the Higher Education Commission of Pakistan for funding this project. I really thankful for the IBBC 2023 for giving me the opportunity to present my research work here. Here I mention my email address. If somebody have any question, please inbox me. Thank you for listening. In that.
Our next presenter is Zohra Naseem from Kohad University of Science and Technology. Our presentation topic is molecular characterization of Babesia species based on cytochrome B gene in large ruminants of district Bajour.
Assalamu alaikum. I am Zohra Naseem, Department of Zoology, Kohad University of Science and Technology, MPhil student. My presentation and research topic is molecular characterization of Babesia species based on cytochrome B gene in large ruminant of district Pajawar. Now start from the introduction. Babesiosis is a tick-borne disease of ruminant equine, cattle, and human brought by an intra-erythrocytic protozoan of the genus Babesia present worldwide but is most common in tropical and subtropical areas. Because Babesia prevalence is high in those areas who have a high temperature. Tropical and subtropical areas also have a high temperature, so that's why this genus Babesia is most common in tropical and subtropical areas. Large-scale mortality and morbidity are caused by it, particularly in developing nations like Pakistan. More than 100 species of Babesia are found around the world, but only two, Babesia bovis and Babesia begimana, are dominating in the tropical, subtropical, and temperate region, affecting millions of livestock population. Pakistan livestock is also considered one of the most important commodity and ranked fourth in milk production worldwide. The current study is designed to find the actual genetic diversity of the Babesia species and to evaluate the parasite species diversity and evolutionary relationship in Khyber Pakhtunkhwa, Pakistan. My research objective to investigate the molecular diversity of Babesia species based on cytochrome B gene in large ruminant of district Pajawar, Khyber Pakhtunkhwa, Pakistan. Come to the material and method. Firstly, sample collection. Approximately 200 blood samples are collected by the help of a syringe from the jugular vein of large ruminant animal in district Pajawar on the basis of clinical signs and symptoms. Samples are stored in EDTA tube and labeled with the host name, month, gender, and age of the animal from which blood was taken, and then transported to molecular parasitology and virology laboratory in the Department of Zoology, Cohort, University of Science and Technology. The collected blood samples were stored at -4 centigrade for further analysis. Then we done a microscopy. About 10 microliter drop of blood placed on a clean slide with the help of a pipette and fixed with methanol. The Babesia slides were stained with Gamson stain and after a few minutes washed with the help of a distilled water. The slides were comparatively dry, then observed under high-power 100x of microscope. Immersion oil was used, which condensed the light and make the slide more clear. This one is a microscopic picture of large ruminant blood. Then I done DNA extraction through Kygene kit, which have a 4-step lysis, washing, then washing, and the last one is elution. Extracted DNA kept -4 centigrade for further analysis. Then I done PCR, polymerase chain reaction, which have 3 steps: denaturation, annealing, extension. Initial denaturation step of 5 minutes at 95 centigrade, denaturation for 45 seconds at 95 centigrade, annealing step of 35 seconds for 54 centigrade, extension phase of 72 centigrade for 45 seconds, and final extension 72 centigrade for 7 minutes. Then I done gel electrophoresis. 1.5% agarose gel is used. The positive PCR samples were sent to the commercial company for the sequencing to find the nearest related species of Babesia. The related match nucleotide sequence were selected. Multiple alignment was done with cluster W. Omega X was used for molecular evolution and for genetic analysis. This one is my study area map, which is Pajawar, the northern area of Khyber Pakhtunkhwa, Pakistan. Results: gender and month-wise prevalence of Babesia begimana. Female have a high prevalence as compared to male, due to the fact that they were kept longer for breeding and milk production purpose, support insufficient feed against their high demands. If we see a month-wise prevalence, July has a high prevalence rate as compared to other months because in July the temperature is high. Age-wise and host-wise prevalence, cattle have a high prevalence as compared to buffalo. If we see age-wise prevalence, adults have a high prevalence rate as compared to the young one. High prevalence were reported in animals aged more than 3 as compared to 1 to 3 age animal because in worse age resistance of the disease were adults show more susceptible than cause, protected by a cholesterol immunity. Area-wise prevalence of Babesia vagina, Salarzai has a high prevalence rate as compared to the Balgina. This one is the microscopic picture and this one is the gel picture. Microscopic prevalence is high as compared to PCR because in microscopy we didn't know what it was, either it is a Babesia or Thalaria. But in PCR we confirmed that this one is the Babesia and which species of Babesia is present, and we know the correct result. So that's why the PCR has low prevalence as compared to the microscopy prevalence. Phylogenetic tree. Phylogenetic tree represent evolutionary relationship among or between a group of organisms and show how species are evolved from a series of common ancestors. For example, Babesia bovis and Babesia ovata and Babesia bejimana both are distinct, but they have a common ancestor. So the phylogenetic tree helps or show the common ancestor. This one is the cytochrome B gene, phylogenetic tree of cytochrome B gene of a Babesia species. Conclusion and recommendation: the highest prevalence rate of Babesia bejimana was found in female host, both cattle and buffalo. The cattle were more effective by Babesia bejimana as compared to buffalo. In month of July, the prevalence rate was observed high. In the middle age, 3 to 6 old cattle and buffaloes are more affected as compared to other lower and higher age group. Similarly, in Salarzai, the prevalence rate of Babesia bejimana was significantly high. The PCR was more sensitive as compared to microscopy. The prevalence measure followed by the owner of the cattle as well as by the government to minimize the prevalence ratio of the babesiosis. These are the references. Thank you. Hafiz.
Our next presenter is Aisha Irshad from Kohart University of Science and Technology. Her title of presentation is Molecular Detection of Microlide Resistance Gene in Staphylococcus aureus isolate from market fish.
Hi, this is Aisha. I am MPhil student from Kohart University of Science and Technology, KPK, Pakistan. I am here to present my research topic. My research topic is molecular detection of macrolide resistance gene in Staphylococcus aureus isolated from fish market. My presentation will include introduction, objectives, material and method, result, conclusion, and finally the recommendation. Staphylococcus aureus is one of the most common bacteria which is found in animals and in humans. But this bacteria sometimes become pathogenic and cause many infections. These infections are treated with antibiotics. Macrolide is also a broad spectrum antibiotic which is used against these infections. It's include erythromycin, clarithromycin, azithromycin, and etc. Some other drugs. But due to their constant and frequently used in treating different humans and veterinary diseases related with the S. aureus, it can cause an emerging case of antibiotic resistance in S. aureus and can cause a threat to the surrounding environment and the contaminated food and the consumer which is consuming it. So it is important to have systematic data about antibiotic resistance in different bacteria, so we can make important strategies to control the transmission of resistant gene to humans or to our environment. The objective of my research is to determine macro-lead resistant susceptibility pattern in isolated S. aureus from the market fish to categorize the macro-lead resistance gene RMA and RMC in S. aureus. The methodology is started with collection of fish samples from different fish markets and then isolation of bacteria S. aureus by growing it on mannitol, salt, agar, and some other important biochemical tests to confirm its presence. After that, antibiotic susceptibility test was be done. So we in which can see easily that which strain of bacteria are resistant or which are not. After the antibiotic susceptibility test, we done DNA extraction of the resistant bacteria or resistant isolates which we get from the fish samples. After that, we run PCR for the amplification of resistant gene, and the result was then sequenced and statistically analyzed. The result of my research is that there are two important genes, resistant genes, which I was looking in as resistant isolates. First one is RMA and second is RMC. As we can see there, the percentage of RMC is relatively higher than the RMA, which is 48.7. So it is can be concluded that RMC is the most prevalent type of resistant gene found in macro-lead resistant bacterial strain S. aureus. The conclusion is that the high prevalence of this resistance, which is a macro-lead resistance found in S. aureus isolate, can be threat to the consumer, which are consuming these fishes, or the humans that are handling them, the fisherman or the market, the seller, the shopkeepers, or the person who is handling them and who is cooking them, and it can also transmit resistant gene to humans. So it is recommended that there should be a continuous monitoring and surveillance on the antimicrobial resistance pattern in organisms, especially microorganisms, so that we can control the spreading of these resistant genes to other animals and humans, because it's make the treatment of some other infection and critical diseases more complicated.
Thank you so much, Aisha Ishad, for your informative talk. Our next presenter is Shagufta Saeed from University of Veterinary and Animal Sciences, Lahore. Her title of presentation is Production of Gallic Acid under Solid State Fermentation by Utilizing Waste from Food Processing Industry.
Bismillahirrahmanirrahim. I am very thankful to the organizing committee for giving me the chance to present in International Brain and Biomedicines Conference organized by Department of Methodology, Government College, University, Faisalabad. The topic that I am going to discuss today is production of gallic acid under solid state fermentation utilizing waste from food processing industries. My name is Dr. Shagufta Saeed and I am working as an assistant professor in the Institute of Biochemistry and Biotechnology, University of Veterinary and Animal Sciences, Lahore. Gallic acid is a phytochemical compound that is present in various parts of plant as secondary metabolite. It is colorless to slightly yellow color and crystalline compound, melting point of 252 degrees centigrade. It has a wide range of applications in pharmaceutical industry as an antioxidant agent, antibacterial, anti-cancer, and precursor of antibacterial drug called as trimethoprim. In food industry, it is used as food preservative. Mainly, gallic acid is produced by hydrolyzing tannin using the acid hydrolysis or enzymatic hydrolysis. On the other hand, some microorganisms have the ability to hydrolyze tannin by producing the enzyme called as tannase. And tannins are naturally occurring water-soluble polyphenols that are present as secondary metabolites in various parts of plants such as wood, bark, leaves, peels, seeds, fruits, and galls. This is a list of various microorganisms that have the ability to produce tannins. Conventional production of gallic acid by chemical hydrolysis of tannic acid is unfavorable due to poor yield, high cost, and discharge of lethal effluent that cause environmental pollution. So the present research was designed with the hypothesis that fruit wastes that are rich in tannin have the potential to be used as substrate for the production of gallic acid by solid state fermentation. These were the objectives of the study: to evaluate the potential of different fruit byproducts that are rich in tannic acid to produce gallic acid by solid state fermentation using Aspergillus niger, and then the optimization of different physiochemical parameters for the production process. Aspergillus niger was first obtained from the first fungal culture bank of Pakistan, University of the Punjab. It was then maintained on Sabarots, dextrose, agar, and preserved at 4 degrees centigrade for further use. Pore suspension was produced by adding 10 ml of 0.9% sodium chloride solution to the plant of fungus until it was further diluted till the spore suspension of 3 into 10 raised to power 7 spores per ml were obtained. It was used as inoculum for the fermentation studies. These were different substrates that were used, like peels of banana, pomegranate, mango, apple, and the fruit seeds of jamun, black plum, apple, mango, and tamarind. Peas and seeds were dried and then ground to a fine powder. Then this powder was stored at room temperature to be used as tannic acid substrate for solid state fermentation. First of all, the tannin content of the substrates were estimated by spectrophotometric method. Then the substrates were screened to achieve the production of gallic acid. Different substrates were taken, 5 grams of each substrate in different flasks, and the substrate water ratio was set at 1:2, pH 4. Autoclaved, then 1 ml of inoculum was added and incubated at 30 degrees centigrade for 72 hours. And then the substrate that gave the highest yield of gallic acid then was further selected for the optimization of different conditions like substrate water ratio, incubation period, inoculum size, and pH, temperature, effect of addition of different carbon sources and nitrogen sources. After the fermentation was completed, then 50 ml of distilled water was added to the flask and kept at orbiter shaker at 150 rpm for 10 minutes, then filtered, and the gallic acid was estimated by mixing with methanolic redonin, and the absorbance was taken at 520 nanometer by using the spectrophotometer. Then the crystallization of gallic acid was done by Soxhlet method. Gallic acid produced was identified by FTIR, and purity was estimated by HPLC using the standard of sigma Aldrich. All the experiments were carried out in triplicates, and the data was analyzed by one-way ANOVA on SPSS software. These are the results of the study. This shows the growth of Aspergillus niger on Sabarots, dextrose, agar plates. Tannin content of different food byproducts, as it can be seen from the table, that jamun seeds had the highest amount of tannin content. Therefore, from different substrates selected, jamun seeds yield the highest amount of gallic acid by fermentation. Then further conditions were optimized using black plum as the substrate. Highest yield was obtained at 1:3 of substrate water ratio, incubation time of 96 hours, inoculum volume of 2, pH of the basal media set at 5, 30 degrees centigrade of incubation temperature. From different carbon sources, glucose gave the highest production. None of the nitrogen sources had the ability to give the highest yield of gallic acid. These are the crystals of gallic acid. FTIR spectrum of gallic acid. The red one shows this our sample, while the blue one is the standard, and it can be seen that similar peak pattern was observed. This is the HPLC chromatogram, and 99% yield, 99% purity of gallic acid was estimated. At the conclusion, different fruit phases were tested for the gallic acid production, from which the highest yield of 13.85 milligram per gram was obtained using the German seeds. These conditions can further be utilized on pilot scale to obtain highest yield of gallic acid for the commercial needs. These are the references of this study. Thanks for your attention.
Thank you so much, Shagufta Saeed, for sharing your amazing input. Our next presenter is Arfa Azim from Kohart University of Science and Technology. Her title of presentation is Whole Exome Sequencing, Identify a Pathogenic FA2H variant in Pakistan family with hereditary spastic paralgesia.
Hi everyone, Assalamu alaikum. Assalamu alaikum. Thank you for giving me an opportunity to present my topic in International Brain and Biomedicine Conference 2023. Starting in the name of Allah, the most powerful and the creator of everything. My name is Arfa Azim, PhD student in the Department of Biotechnology and Genetic Engineering at Kohart University of Science and Technology. My topic of presentation is the whole exome sequencing identified a pathogenic FA2H variant in Pakistani family with hereditary spastic paraplegia. The contents of my presentation is the introduction, why we choose this study, objectives, material methods, result data, and references. First, I will give you a brief introduction of the hereditary spastic paraplegia. As we know that there are more than approximately 25,000 genes which are reported for the human and within the human body, out of which 10,000 genes are known to be reported for the brain morphology and physiology, which is a very huge part which is responsible for the nervous system. And also we know that the nervous system is the most complicated system of the body, which controlling all other systems of the body. And now if some mutations occur within these genes, so it will cause the neurological disorders, out of which hereditary spastic paraplegia is also one of the rarest neurological disorders, which having the phenotypes of poor coordination of hand, speech, and eye movement, and also the gait abnormalities, which is a major phenotype of the hereditary spastic paraplegia. And also there is a spasticity of muscles occur within the spastic paraplegic patients. Now, why we choose this study is we know that the neurological disorders are very and having the mixed phenotypes, and also there is a high clinical and genetic heterogeneity, which make it difficult to diagnose at clinical level. So we analyze it genetically to specifically diagnose these type of disorders. And also the unavailability of cost-effective diagnostic feasibility, unestablished molecular diagnosis of the patients with hereditary spastic paraplegia in Pakistan. The fourth one consequences is there is a high rate of consanguinity, which is about 70% within the Pakistani family. So it causes to spread the such type of disorders within the Pakistani population. So to overcome these consequences, we develop some objectives, which is to identify a causative genetic variant within the causative genetic variant in patients with hereditary spastic paraplegia. To determine the disease-causing alleles, segregation in the family members of patients. Third one is to predict the pathogenic effects of genetic variant on protein structures and functions. To overcome these objectives, to achieve these objectives, we follow some methodology. First, we obtained ethical approval from the CUST Ethical Committee at Cohort University of Science and Technology, and also we signed up a consent form from the affected family members. Second, we extracted genomic DNA and also direct sequencing. First, we prefer the direct sequencing of some common genes, which is responsible for the hereditary spastic paraplegia within the specific population. But if we didn't find by direct sequencing, so then we further goes to the advanced technologies like next generation sequencing, microarray genotyping, data analysis, and co-segregation of genetic variants within the families. Fourth one is to predict the pathogenicity of genetic variants and also mutant protein structure prediction within the affected family by the help of online available bioinformatics tools and software like Phi2, Enfold, Mutation Tester, etc. The results of this study is here you can see that the families consist of the 4th generation. Clinically, this was diagnosed as a hereditary spastic paraplegia. Phenotypes of this family member is the limb weakness occur in the affected member of the family, and also there is walking abnormalities and poor coordination. For the pedigree analysis, you can see the key at the right side of the slide. After the whole exome sequencing, we received the VCF file. Then it was converted by the online softwares to the annotated file. For further analysis, we applied these different types of filters on the annotated file. First one filter is applied for genotyping, and the second one is to identify the numeric frequency of the genetic variant. Third one is for coding effect, in which we selected missense plus frame shift. The fourth one is for applied variant location effect, in which we mostly choose the exonic plus splice site region. Fifth one filter is applied for the HGMD phenotype, which is the hereditary spastic paraplegic phenotype. Finally, we identified a FA2H mutation in FA2H, which is the deletion of codons occur from the 159 nucleotide to 167. There is a deletion of nucleotides occurs, which affected the protein structures and protein at the position of arginine 53. 5 codons were deleted, so it affected the protein structures and also the function of the protein. So for further analysis, in silico analysis was performed, in which we use different online bioinformatics tools like the mutation tester, and also we use the Versome, and for the identification of the numeric frequency, we also use the numeric Genomed software. Here is the summary of this presentation. First, we looked at the family, then we signed up the consent form, and also obtained the ethical approval for this study. Then we analyzed the family, genetically analyzed the family. So we find out the FA2H genetic variant and the FA2H gene, in which deletion of many nucleotides occur, and also which affected the protein and structures and functions, which is protein structures and functions. Also, you can see that the complete segregation of this variant within this family. Here you can see that the mother of this patient is heterozygous for this genetic variant, and also the one sister is a heterozygous carrier for this genetic variant. One brother is heterozygous carrier for this variant. The two sisters, the first one and second one sisters are normal for this genetic variant. So here is the differences of this study. Thank you for questioning. For questions, you can contact me with this email, through this email. Thank you. Thank you so much.
Thank you so much, Arfazim, for sharing your knowledge with us. I would like to thank everyone for your participation. Next session will be continued by my another colleague.